The role of O26 polysaccharides in immune evasion of enteropathogenic E. coli and vaccine development. Introduction: Escherichia coli, E. coli. O26 is a group of bacterial strains that includes enteropathogenic E. coli, EPC, and enterohemorrhagic E. coli, EGC, both known to cause severe gastrointestinal diseases. EPAC is associated with childhood diarrhea in developing countries, while EHEC can cause bloody diarrhea and hemolytic uremic syndrome (HUS) globally. An important component of these bacteria is the O26 polysaccharide capsule, which plays a crucial role in immune system evasion and vaccine development. Structure and function of O26 polysaccharides. O26 polysaccharides form a capsule around EPC strains identical to the O-antigenic polysaccharides found in their lipopolysaccharide (LPS) molecules. These polysaccharides are essential for the bacteria as they help protect against the host's defenses, especially the complement system, which is a fundamental part of the innate immune response. The capsule is classified as a group 4O antigen capsule, synthesized via the WZX/WZY pathway. Immune evasion: the EPEC polysaccharide capsule prevents the deposition of C3b, a protein of the complement system, on the bacterial surface. This action protects the bacterium from complement-mediated lysis and phagocytosis by macrophages. The study reveals that the EPEC capsule contains high levels of glucose, heptose, and N-acetylglucosamine compared to EHEC, which contributes significantly to its ability to evade immunity. Comparison between EPEC and EHEC. Chromatographic analysis showed significant differences in polysaccharide composition between EPC and EHEC. In EPC, the proportion of glucose, heptose, and N-acetylglucosamine was much higher compared to EHEC. This difference in composition may explain the variations in immune evasion capacity between the strains. While EPEC has a thick capsule that prevents the deposition of C3b, EHEC, which does not have a similar capsule, is more vulnerable to the host's immune response. Humoral immune response-specific antibodies against O26 polysaccharides were able to neutralize the protection conferred by the EPEC capsule. These antibodies inhibited EPEC adhesion to human epithelial cells and promoted bacterial lysis, proving effective in recognizing both EPC and EHEC O26, but not other E. coli strains. This specificity is crucial for vaccine development, as it allows a targeted response against pathogenic strains without affecting commensal strains. Detailed experimental results. 1. Visualization of the capsule. The Manifold staining technique showed a thick capsule in EPAC O26, while EHEC O26 showed no capsule. This result was visually confirmed by the presence of a transparent halo around the EPEC bacteria, indicating the presence of the capsule. 2. Gas chromatography analysis. Gas chromatography revealed higher levels of glucose, 1.69, heptose, 3.6:0.89, and N-acetylglucosamine, 4.5:2.10, in EPC compared to EHEC. This differential suggests that the EPEC capsule is composed of a more complex structure, contributing to its evasion capacity. 3. ELISA assays. Antibodies against O26 polysaccharides recognized EPEC and HEC O26, but not unrelated E. coli strains such as E. coli O127 and E. coli DH5 alpha. This assay demonstrated the specificity of the antibodies, which is crucial for the development of specific vaccines. 4. Bacterial lysis. The complement system was able to lyse non-capsulate HEC and E. coli DH5 alpha, but not capsulate EPC unless anti-O26 antibodies were present. This was determined by a bacterial viability assay after incubation with normal human serum. 5. Phagocytosis. The presence of anti-O26 antibodies facilitated the phagocytosis of capsulate EPEC by macrophages. Macrophages incubated with capsulate EPEC and normal human serum were unable to phagocytose the bacterium, but the addition of anti-O26 antibodies reversed this resistance. 6. Adhesion to epithelial cells. Anti-O26 antibodies inhibited the adhesion of capsulate EPEC to HEP2 epithelial cells. This result was observed through adhesion assays where the presence of anti-O26 antibodies significantly reduced bacterial adhesion. Discussion. O26 polysaccharides play a crucial role in EPEC immune evasion, mainly by inhibiting C3b deposition and phagocytosis. However, the presence of specific antibodies against these polysaccharides can neutralize this protection, making them promising targets for vaccine development. The research highlights that an effective vaccine against O26 polysaccharides could prevent initial bacterial adhesion, promote bacterial elimination, and offer protection against infections caused by these pathogenic strains. Implications for vaccine development. The development of vaccines against E. coli O26 strains targeting O26 polysaccharides could provide robust protection against infections. Antibodies generated against these polysaccharides have been shown to be effective in neutralizing the protection conferred by the capsule, facilitating the host's immune response. In addition, a targeted vaccine could prevent interference with commensal strains of E. coli, which are beneficial for human gastrointestinal treatment. Conclusions. O26 polysaccharides represent promising antigenic targets for the development of vaccines against capsulate strains of E. coli O26. These vaccines have the potential to elicit an effective humoral immune response, counteracting immune evasion mechanisms and preventing infection. This research provides valuable insights for the development of immunization strategies against EPEC O26, highlighting the importance of O26 polysaccharides in bacterial pathogenesis and immune response. Perspectivas. Future investigations should focus on the detailed characterization of the mechanisms regulating capsule expression in EPC and EHEC, as well as clinical trials to evaluate the efficacy and safety of O26 polysaccharide-based vaccines. Furthermore, additional studies on the interaction between O26 polysaccharides and other components of the immune system may reveal new strategies to fight bacterial infections.